So my name is Sara, and I am Product Manager and Scientific Support Specialist at Takara Bio Europe, and responsible for the NGS product line. So if you have any questions during this webinar, please type it in the chat box and we will take them at the end of the presentation. Today I will start talking to you about how we have enabled doing single-cell RNA-Seq using the SMART technology. First, I'll discuss what are the current challenges of single-cell RNA-Seq and how our SMART-Seq before technology has allowed to cope with these challenges. Then I will discuss how this technology can also be applied to other challenging samples like degraded RNA or ultra-low input RNA, small RNAs, and T-cell receptor profiling. And I will finish by briefly discussing some applications of this technology to DNA. So before we start talking about single-cell sequencing, a question that comes often is what's the interest of doing this? Why would someone would like to study single cells? So it's been shown since some years now that actually gene expression is very variable between different cells. And when you are taking a population of cells, a bulk population, and you extract RNA from that, actually what you will see is an average expression of the population, which not necessarily will correlate with the expression of each single cell. So here we see an example of this in this paper in which the authors were interested in evaluating different genes in a bulk population, here 10,000 cells, or different single cells, here shown in blue. So you will see that there are two types of genes depending on their pattern of expression. So for these genes here in the left, you see that the pattern of splicing and expression is very similar between the bulk population in gray and each of these single cells. However, they identified another group of genes for which there were differences between the patterns. So you can see here for this gene that in the bulk population you have a very constant pattern. Then if you see all different single cells, you will find differences in the splicing pattern and in the level of expression. So this is only an example. There are now many studies showing this kind of differences. And what's important to know is that these cell-to-cell differences may be critical in many physiological processes, but also in disease. So working with single cell, and especially being able to do RNA sequencing from single cell, is very challenging. So first, we can think about the difficulty linked to the fact that we are starting with tiny amounts of RNA. So you can estimate that a single cell have around 10 picogram of total RNA. And of this total RNA, only around 1% will correspond to actually mRNA, so the fragment of the RNA population that would actually encode protein. So if we want to do single-cell RNA sequencing, we need an extremely performant method that first needs to be sensitive to be able to do this cDNA synthesis from this extremely tiny amount of RNA. It needs also to be reproducible, and this is very important because we want to analyze biological variation, and if we have a high technical variation, we will be masking these differences in which we are interested. Of course, it's important that the method is able to take cells as direct input, because you can imagine that extracting RNA from one single cell is nearly impossible, I would say. And for sure, we want also to reduce the handling to avoid the risk of degradation, of contamination, because working with RNA is very sensitive, as you may know, and also to limit sample loss, since we are starting with such a small amount of material. And finally, you may also want to obtain a full-length cDNA in such a way that you will maintain the transcript length and the representation of your different transcripts in the original RNA pool. So this is actually what we are at Takara Bio developed and we offer here with our technology, which is called SMART. So I'm going to explain here briefly how does it work, the SMART technology, for the case of our product for single-cell RNA sequencing, which is called SMART-Seq V4 ultra-low input RNA kit. So here we have the description of the workflow. So actually, SMART is an acronym for a switching mechanism at the 5' end of the RNA template. And it is based in two properties of the reverse transcriptase that we use to do the cDNA synthesis that I will describe here. So we start the reverse transcription with an oligo. In this case, it's an oligo DT, which also contains a known sequence, here shown in green, called the SMART sequence. And then the reverse transcriptase will do the reverse transcription. And when it reaches the 5' end of the template RNA, it will add some nucleotides, non-template nucleotides represented here as X's. And then we have characterized this specific addition of nucleotides, and we have designed an oligo called template switching oligo, which will contain bases that anneal to this non-template nucleotide. And also will carry another stretch of this SMART sequence, again here in green. So after the annealing of this template switching oligo to these non-template nucleotides, the reverse transcriptase will change the template. So this is what we call template switching. And it will change from using the RNA as template to use this oligo, the SMART oligo, as template. And then we'll continue the reverse transcription, and as a result you will have a first-strand cDNA which contains known sequences in both 5' and 3' end. Now we can use these sequences that we know and that we have incorporated as annealing sites to use a primer to do a PCR and amplify the full-length cDNA. So basically what this is allowing us is to first we will be able to work directly with cells because it's a very sensitive technology. And of course you can also, if you have purified RNA, you can also use it. So the input of this kit is 1 to 1000 cells or 10 picogram to 10 nanogram total RNA. So as we are using here oligo DT, we do not require any ribosomal RNA removal or a DNase treatment because oligo DT will selectively amplify only poly-A RNA. This also implies that if you are starting from total RNA, your RNA needs to be of high quality. So for this technology, we recommend a RIN of at least 8 or higher. All these processes is very fast. It's in a single tube, so limiting the risk of material loss and contamination. And at the end, we will have a full-length cDNA. As we can see here with this bioanalyzer trace profile, this is the typical result that we observe from human cells. We will have a distribution between 400 and 10 kb for human cells, and from different input amounts is very reproducible. So this full-length, very long cDNA is not suitable directly for sequencing. We need first to do a library preparation, so it means to cut it out in pieces suitable for sequencing and add in the adapters. So the process is different if you are using Ion Torrent or Illumina. If you are using Ion Torrent, we provide a protocol which will use Thermo Fisher reagents for enzymatic shearing and library prep. If you're using Illumina sequencing, you have two options, so it depends on what's your preferred method. You can either do a mechanical fragmentation using Covaris and then use our low input library prep kit to add the Illumina adapters. Or you can do a modified Nextera XT protocol, and when I say modify, it's the classical protocol, is just that it requires less input amount. And Nextera XT protocol, what it does is it will do fragmentation. It means it will fragment the cDNA and will add the adapters for sequencing. So of course you can do that manually, let's say, but there is also a version of this technology which has been developed for this instrument, which is called the Fluidigm C1, developed by another company called Fluidigm. And what it allows is to automatically load and capture single cells in different wells of a microchip, and it will add all the reagents that are required for the reverse transcription and cDNA amplification using SMART-Seq v4 reagents. So as you have noticed, then the name of this kit is the version 4, and meaning that we have had other versions in the past. And indeed, we strive to continuously improve the technology to provide the most sensitive results. And in this case, in the version 4 of the technology, we did some changes, but one of the most important was to incorporate LNA, which stands for log nucleic acid, and also optimizing the template switching reaction. And this, what allows us is to dramatically improve the sensitivity and also the reproducibility, as well as reducing the background. And how do we see that? So here we have a comparison between our previous versions, so the ultra-low version 3 and SMART-Seq v4, only using 10 picogram of mouse brain RNA. So you remember that 10 picogram is roughly the equivalent of the content of RNA in one single cell. So what we see here is that with SMART-Seq v4, we are able to use less cycles of amplification because it's more sensitive. And even doing that, we are identifying between 30 and 40% more transcripts than the previous version, as you can see here. The reproducibility is very good. It was already the case here in the previous version. And we kept this also in SMART-Seq v4. But as I say, we also improved on that. And how do we see that? We see that especially for the low expressed transcripts. So here we are comparing the results obtained in 2 replicates, again with 10 picogram of RNA, using the 2 technologies, the version 3 and the version 4. So if we measure the overall coefficient of correlation between the 2 experiments, we see that correlation is quite good for the 2 technologies, because we are closer to 1, but we see that it's better here for SMART-Seq v4 than for ultra-low v3. But this difference is even more pronounced when we analyze this part, the lower part of each chart, because here we will have the transcript that are expressed in a lower amount, so those which are more rare. And if we only zoom in this part, what we see is that the coefficients of correlation with the version 3 were below 0.4, while with SMART-Seq V4 we are above 0.6. So, and you can also see that the points are much more closer here as compared to here. So this result shows that we can be confident with the results obtained with V4, even for transcripts that are very low expressed. Another parameter that we measure is the gene body coverage, and for this particular experiment we use single cells. As you can see here, 6 different single cells, or a bulk population of 100 or 1,000 cells. And what's interesting to note here is that whatever the input that we use, single or bulk population, we see a very even gene body coverage with SMART-Seq V4. So these are some only some examples of the data that we obtained with this. We have many more on our website. So before finishing this part with the single cell, I would just like to let you know that we have developed novel applications using this technology. In particular, this technology called the SMART-Seq V4 3'DE, and DE stands for differential expression analysis. So this is useful if you are interested only in measuring differences of expression between different cells or different populations, and you don't need the full-length cDNA, because for doing this differential expression analysis, only capturing the 3'N is sufficient. So this is what it allows this kit. So you can see here that as compared to the image that I showed before, the coverage is only on the 3'N side. So this, of course, will allow you to do differential expression at the single cell level at a lower cost. Another application of SMART-Seq V4 technology is target RNA sequencing. So let's say that you're interested only in studying some specific transcripts. You don't need to amplify the whole transcriptome because you have a family of transcripts that you are interested in, or you are interested in studying a specific splicing isoform or in detecting a gene fusion. So these are events that are very rare, and if you amplify the whole transcriptome, it will be difficult to identify them. So what you can do is first just to capture your transcript of interest, and then do library prep only from these transcripts of interest. And this is what this smarter target RNA capture will allow you to do, right? So this technology has been already out for some years now, so from 5 years. And since then, there have been many high impact papers that have shown the results that you can obtain with smarter technology, especially in single cell. It has been used with many mammalian models, as they are listed here. The methods to get the single cells can vary. You can do it manually. You can use FACS sorted cell. You can use laser capture microdissected cells. You can use the fluid IMC one, as I mentioned. It has also been used with non-mammalian, so that's important to know, because maybe you are not working with mammalian, maybe you're working with another eukaryote. So the kit will work with any eukaryote material, because as long as it is polyadenylated, it should work. So what you can't use with SMART-Seq V4 is two different inputs. So the first is low-quality RNA, because if your RNA is degraded, normally the RNA degradation starts on the extremities. So a degraded RNA has lost usually its poly A tail. If there's no poly A tail, there is no reverse transcription with the oligo DT primer. The second is, of course, samples that are not naturally polyadenylated. So it means prokaryotic RNA, which usually is not polyadenylated, is not suitable for this technology. So this brings us to the second part of the presentation, which is what to do if you have degraded RNA or non-polyadenylated samples. So for these cases, we have adapted the technology. So you will recognize here the workflow is very similar to what I already showed before. It's just that we are well, we incorporate some differences to make it compatible with degraded or non-poly RNA. So the first one is that we are going to use N6 primer, so it means a random primer or random hexamer as you call it, to prime the reverse transcription. So this will allow us to start with RNA of any quality. So we have tested this kit with a ring from 3 to 10. So it allows to work with degraded RNA, FFP samples, laser capture microdissected samples, and so on. So here also we have modified the technology to incorporate in the same time during the PCR steps the Illumina indexes and adapters. And this has a great advantage is that you don't need to do downstream library preparation, and it makes it very fast. It means that you will start with a degraded or fragmented RNA, and you will end up with a library ready to sequence in a few hours. So something that is important to note here is that the input for this kit is a ribosomal RNA depleted RNA. So as you may know, when you are doing total RNA-Seq, so using random hexamers, you will amplify everything, including the ribosomal RNA. But you don't want to keep this, or you don't want to do that, because ribosomal RNA will capture around 90 to 95% of your reads, and it's not usually the information in which you are interested. So you will need first to treat the input RNA with a method to deplete it from the ribosomal sequences. We at Takara, we have a product for doing that from mammalian sample, which is called Ribogon. And this Ribogon is compatible with as low as 10 nanogram of total RNA. If you are working with other species and you are using other methods to deplete your ribosomal RNA, you can use directly the smarter stranded RNA-Seq kit. And in that case, the input is very low. So with as low as 100 picogram of depleted material, you can start. Okay, so what happens if you have less than the 10 nanogram that is required to do ribosomal depletion with Ribogon? So this is quite challenging because at the moment there is no commercial solution to be able to remove ribosomal RNA from such low input amount. So at Takara, what we did is that we took the problem differently. Instead of degrading or trying to remove the ribosomal RNA, we say that we are going to prepare a library from all the RNA, including the ribosomal RNA, and then at the once that we have the cDNA, we will only target ribosomal cDNA sequences. And this is what this kit smarter stranded pico input mammalian allows. So you will be able to start with as low as 250 picogram of total RNA, and then you will do reverse transcription, cDNA synthesis, amplification, and using a proprietary technology, you will be able to target ribosomal cDNA sequences, so using what we call ZAP-R and R probes, and degrade those, and then you will be able to enrich for your library containing your fragments, your non-ribosomal cDNA sequences. So this Pico kit is also compatible. It's using random priming. It's compatible with also very degraded RNA. You can go as low as 2, and you will have all the advantage of the stranded technology. It means that it's very fast. You will get a library ready to sequence. And maybe something that I didn't mention when I talked before about the smarter stranded kit is, of course, as the name indicates it, this technology allows to keep strand information, and this can be important for people who are studying maybe antisense RNA or non-coding RNAs, or this kind of information. We have here some metrics showing the performance of the Pico kit. We measure different input amounts, so the higher, which is 10 nanogram, up to the lower, which is 250 picogram, and even lower than the recommended input amount. So you see here that we use it with as low as 100 picogram. What's interesting to see here, and each time we have 2 replicates, that the results are very reproducible. The number of transcripts identified is high, no matter of the quantity used. Of course, when you are working with such low input amount, the metrics are starting to be less good, so you will have a high duplicate rate. So that's why, I mean, it's the choice of the customer, but we recommend to stay with at least 250 picogram to have the best results. Something that you may notice here is the percentage of ribosomal RNA that is detected after in these Pico libraries. So if we compare to Ribogon, so the technology that I mentioned before, Ribogon is a very efficient technology. You will end up with only around 1% of ribosomal RNA. Here you will see that we are rather around 10% of RNA. So this means that the technology that we use here to deplete ribosomal cDNA is less efficient, right, than the technology used in Ribogon. But still, what this 10%, let's say, is much better than the 90% that we would find if we don't do any treatment. So this can be also important when we are working with very more challenging samples like FFPE, which are degraded, and also depending on the tissue, sometimes the level of ribosomal RNA can be very high. So here we took a human liver total RNA FFP sample with a reading of 2.5, and this is a very complicated sample because even when started with the high input amount, we are always around 30, more than 30% of ribosomal RNA left, as you can see here. So, but what's interesting is to compare what happens if we don't use the method, the ZAP-R, on the R probes to deplete ribosomal RNA. And what you can see is that if you don't do anything, you will have 90% of ribosomal RNA. So getting 30% is already a gain, but more importantly is the number of transcripts identified. You will only identify a third of the transcripts if you don't do this as you identify when you perform the procedure. So the Pico kit will allow you to capture small size fragments, so at least 100 nucleotide of size, as you can see here from this curve. But if you need go even lower because you are working with small RNA, so less than 100 nucleotide. So this is again a challenging application to get a library from small RNAs, and we have developed a technology for doing that, smarter SM or small RNA-Seq for Illumina. So this kit works differently from what most of other kits in the market use. They are mainly based on ligation to capture these small RNAs, and this has been shown that has some bias, as we'll see later. Basically, our technology again use the same, you will recognize the SMART protocol already described. And this has some advantages, is that you can start with total RNA or with an enriched small RNA fraction, but it's not necessary to enrich the small RNA. And the input is very low, so what we like Takara, as the name of the presentation says, to allow you to work with the most challenging samples. So you will be able to work with as low as 1 nanogram of total RNA and get a small RNA sequencing library from it. We are not requiring with this protocol to do this tedious page gel purification, in which you need to run a polyacrylamide gel and let it run and then cut the small band, all this cumbersome and long procedure. We are providing two options for the library, for the size selection. One is using the blue pippin instrument, the other is doing that by using ampere bits. And the incorporation of the Illumina adapters is ligation free, so we are not having this bias issue, and of course the protocol is very fast. Another advantage of this protocol is as it's selecting small RNAs by size, so you will capture anything between 15 and 150 base pairs. It's very useful for other applications that will generate small or short size RNA fragments, like RIP, CLIP, or ribosomal profiling. So I just said that one of the issues of ligation methods is the bias issue. So it means that there are some microRNAs which are ligated to some adapters more efficiently than others. So this is something that has been very well described, and there are publications on that. And we wanted to see how our method compared to this adapter ligation. So here we took a synthetic library of more of 900 microRNAs, which they are all at the same initial concentration. So here in the X you have all the different microRNAs, and they are all at a known concentration, which is exactly equivalent, exactly the same. So if all microRNAs were ligated with the same efficiency, we should see that the expression level observed as compared to the expression level that we know that we put and is equivalent should be equal to 1. So we should have here just a line around 1, just like here, on horizontal line 1. But in reality, what we observe is that you can see that not all microRNAs will be found with the same expected concentration. And what we see here when using the adapter ligation method is that only 22% of these microRNAs will fall in this twofold cutoff of the expected expression level. The SMART technology, on the other side, will capture 55%. So it means that we are improving these results for 2.5 times. And overall, it will show that the SMART technology will allow you to have a more accurate portrait of the expression of this population of microRNA. Again, we are measuring the reproducibility here between two replicates, 100 picogram here, but even between a tenfold difference of input amount when we compare here 100 picogram to 10 nanogram. So the last challenging sample that I'm just showing here is the T-cell receptor profiling. So this is a specific application as compared to what we discussed before that will allow you to do whole transcriptome analysis. Here we are focusing only in one specific transcript, and this is the transcript coming from the T-cell receptor, alpha and beta. So for people who are interested in this application, we have developed or we have adapted again the SMART technology to be able to do a library only from these TCR alpha and beta sequences. It works with human samples and mouse samples, and you will be able to get the full-length variable sequence of these TCR alpha and beta mRNAs. You can also obtain both alpha and beta sequences from the same sample, I mean the same experiment if you wish, or you can just do it separately as you want. But other technologies you are obliged to do it separately. Here you will be able to do alpha and beta in the same amplification. Also, the input is flexible. You can start with RNA if you decide, or with T cells directly. So if it's human or mouse, you can also do that. And we are incorporating Illumina indexes and adapter again during the workflow. So you start with your RNA or your cells, you end up with a sequence with the library in a few hours. And my last slide is just to briefly mention that we have expanded the applications of the SMART technology to DNA, because it's interesting to know that the reverse transcriptase that we use for all the SMART RNA-Seq methods actually is able also to reverse transcribe DNA. And this is what we see here. We are using the same RT, but to be able to prime this single-stranded DNA, we need first to add a poly-T tail, and then we will use oligo-DA primer. So kind of the opposite that we do with RNA. But the consequence is the same. It means that we will be able to copy this DNA strand to incorporate the adapters and indexes thanks to template switching and to PCR steps. So this technology is available in two different or for two different applications. So the first is for ChIP-Seq, and this technology is very well suited for ChIP-Seq because usually from ChIP what you obtain is DNA that is single-stranded and of very low input amount. So 100 picogram is the minimum input amount for this kit, so it makes it very well suited for this application. It doesn't matter if you are starting with double-stranded DNA or single-stranded DNA. You will have the ligation-free addition of Illumina adapters in only 4 hours, so this is very fast. The same technology has been used for more recent application, which is in this kit, Explore Meta-Seq DNA. That basically what will allow you to do is to first separate methylated DNA from unmethylated DNA using a column also developed at Takara called CaptureM. So this is only a 2-hour protocol that will allow you to separate the two DNA fractions, and then you will apply this protocol, which is also very fast. So in 1 day you can get a library to study global methylome profiles. So this is the application of this kit. So I have shown here just a few examples of the different applications and data of our products, but I would like to invite you to go to our website. We have this NGS resource portal, which is a very complete site with tech notes where you will have all the data, the material and methods that we show. There are webinars also for each of the kits if you want to know more. For example, about TCR, I just showed one slide, but here you will have a detailed explanation about each kit. We have FAQs, citations, and a nice selection guide to allow you to determine which is the most adapted product to your experiment. And of course, if you still have questions, you can contact us. Here is our support address. And this is what I had today to present. And now we are going to take the questions. So please do not hesitate. Okay, let me check this. Okay, so there's a question if the small RNA-Seq can be used with RNA from any species. The answer is yes. Here there's no, something that maybe I didn't say, this protocol starts with a polyadenylation step. So because small RNAs or microRNAs specifically are not polyadenylated, so what we are doing is first doing a polyadenylation. It means that even if you are starting with prokaryotic RNA, which is not poly-A, you will be able to use it as input, because again you will just add this poly-A and then do a oligo-DT reverse transcription. Okay, another question. Is it possible to use a SMART-Seq V4? So this is the single cell kit with frozen cells. So yes, the kit is compatible with frozen cells. We provide some specific recommendations on how to do that. For example, you need always to conserve the cells in PBS. We do not recommend using RNA later or this kind of fixatives, because they are not compatible with the subsequent cDNA synthesis steps. But if you need more information, we can send you the specific recommendations for working with frozen cells. Right. Another question is if we can use SMART-Seq V4 with bacteria for single-cell bacteria RNA-Seq. So no, again, this is this kit uses oligo-DT priming, so you will need poly-A RNA. But the smarter stranded kit has been used. There are many publications on that. We have used it in bacteria samples, and it's been used a lot in metatranscriptomic studies, either from human gut samples or from samples from ocean bottoms, things like that, in which you will have a mixed population. So you can use a smarter stranded for that. Okay. Is there any other question? Right. So if you have any other question, please do not hesitate to write us to this email address. And thank you very much for your attention. Bye-bye.